And I really want to thank the organizers of glycogen seminars for giving me the opportunity to tell you a little bit about myself, a little bit about my lab, what we do, and hopefully some of you will be able to collaborate together. You'll contact me and we can start a conversation together. So just before we get started, I think Mike gave an introduction to me. I want to just, this is my first time talking at Glycogen, so I want to just give you a little bit information about where I'm from and what has brought me to Glycoscience. So let me see how I can. Okay, so I'm going to tell you about my glyco journey a little bit. This year is a special year for me, so I just made this slide and I kind of go through it every time I speak this year because it's my 30th year being at the CCRC. So just starting off, I was born in Iran, and then I immigrated to United Kingdom in 1978 with my family. I did my undergraduate study at University of North London, and then a PhD at Imperial College London in mass spectrometry. I worked for a couple of years in industry before joining the CCRC in 1994 as a postdoc, and I joined the DOE Center for Plant and Microbial Polysaccharides, and that's the picture of the building where we were in when I joined in 1994, which is just down the street from here. I started being involved in service and training, and the picture here is our current training lab that we use for training glycoscientists in different areas of carbohydrates. Later, we moved into the new CCRC building in 2003, and in 2001, I became the technical director of service and training. But all through these years, I was just going along doing my service and training and some research until really in 2020 that really things kind of took off for me with the SARS-CoV-2 research. And then I also became the PI on this NIH Glycoscience resource. Really, our research with the SARS-CoV-2 glycosylation and other spike protein and other proteins there really give us the visibility and put us really on the map. Later on in 2022, things became kind of full circle for me when I became the PI on the DOE Center for Plant and Microbial Polysaccharides, where I had started as a postdoc in 1994. And then later also in 2022, I was able to get the Society for Glycobiology Distinguished Service Award. And now I'm also involved with, as Mike said, BioGrade and an SF grant that has a lot of capabilities for using glycoenzymes and glycoanalytics, high throughput glycoanalytics and AI to look at protein glycosylation. And I'm also involved in Glycomip, which is really trying to synthesize and characterize glycomaterials using different tools. So with that, I'm going to give you some information about what we do at the CCRC as a whole and what my lab does. CCRC scientists and faculty are involved with research, and their information that they obtain kind of pours into my lab. So a lot of the research that goes on at the CCRC is kind of pours into analytical service and training laboratory. My laboratory itself also does research. We do tool development for structural characterization of carbohydrates. So we have a service component, which is a fee for service, and then we do a lot of collaborations with universities, research institutes, and also pharmaceutical and biotech industry. As you may have heard of, we have had extensive training programs at the CCRC. This year is going to be our 40th years of offering training courses, both hands-on and more recently virtual, in glycoscience. And I'll tell you a little bit about that later. So what do? We basically analyze anything that has a carbohydrates attached. So, for example, we do a lot of microbial polysaccharides, lipopolysaccharide, capsular polysaccharide, structural characterizations. We do glycoprotein, viral glycosylation. We've done a lot of fungal glucans. As I said, I'm the PI on the DOE Center for plant and microbial. So we do a lot of plant cell wall polysaccharide tool development and characterizations. Since the last 15 years, my lab has been involved with glycosaminoglycan analysis, heparin, low molecular weight heparin. We were involved with the figuring out the source of contamination about 2007, 2008, when there was heparin, inadulterated heparin, and about over 80 people died. So we were involved with that study starting in 2007, 2008. We do a lot of vaccine structural characterization and glycolipids. We use an array of techniques from different LCMS, mass spec methods to 2D NMR, composition linkage, SAX HPLC. We do a lot of isolation and purification, and people don't usually put that much thought into how important that first step is. Because if you don't get enough of the material you're isolating, or you don't isolate the right material and some of it is left behind, you don't get a good picture of what is the glycome or what are the glycoconjugates. So my lab is really putting a lot of emphasis on tracing the carbohydrates that is coming out of cells or tissues to make sure that our isolation and purification procedures are complete for the type of classes of carbohydrates we're looking at. So eventually we want to look at the structure, and hopefully with those structure information in hand, we can help our collaborators to tackle function. So as I said, we have an array of methods. We can measure the molecular weights of the polysaccharides or oligosaccharides using size exclusion chromatography or mass spectrometry. We do a lot of composition analysis where we look at the monosaccharide constituents that are present in glycolipids, glycoproteins, or polysaccharides through hydrolysis and then detection by different methods. As I said, we do size exclusion chromatography. We do a linkage analysis using methylation analysis and GCMS to look at the way polysaccharides or oligosaccharides are linked. Is it 2-3 linked? Is it 4-6 linked? And what is the relative percentage of those linkages? When it comes to glycoproteins, we can release the glycans and derivatize them and look at the oligosaccharide distribution. We can do site mapping or do glycopeptide analysis where we look at the peptide and the glycans attached. My lab also does quite a lot of NMR analysis. I won't go into much NMR today because I want to concentrate on mass spectrometry methods, but we do a substantial amount of NMR, especially with our new 1.1 gigahertz NMR at the CCRC. This is the NSF-funded grant that has brought the 1.1 gigahertz NMR for liquid state NMR at the CCRC. So we are very excited on employing that technique for better resolution and sensitivity for a lot of our polysaccharide analysis. So I'm just going to give you some examples of what does different data that we get, and I'll go into SARS-CoV-2 as an example of how we've gone about characterizing glycoproteins. So when we collaborate with our collaborators, they could have they may be interested in glycoproteins, they may be interested in glycolipids or glycosaminoglycans. When we start with cells or tissues, we separate these components, and then later on, if the collaborator wants to look at these classes, we can take a look at that. This is an example of where we have released all the glycans from cells, and this is a wild type cell. And what we do initially, the MOL-DMS or the electrospray analysis, this is a case of MOL-DMS, gives you compositions of all the types of N-glycans we can see. And the signals can also give you the relative ratio of these glycans that are present. Later, we can start sequencing those glycans using MS-MS. This is an example that we went to the MS2 and then MS3 in order to detect, for example, bisecting glucanacs. Bisecting glucanacs have been shown to be important biologically, so their presence and their relative amounts are important for a particular biological questions. We also can take a look at some other structures, for example, the presence of 2-galactose residue. This is called alpha-1,3-gal gal-gal residues. These have been, for example, implications for tick bites. And this is an example of saliva from ticks and presence of these structures on tick bites. We've done quite a lot of analysis on ticks and tick fluids, and this is an example of how we determine that structure being present on end-linked glycans. So once we have that kind of profile, we can begin to do a comparative glycomics analysis and get the relative quantitation of one sample versus another, for example, diseased versus healthy patients, cancer cells, knockout cells versus wild types, and really try to answer biological questions regarding the glycans present on the surface of these cells. So then once we have that data, we can start interrogating the data more deeply, looking at the percentage of, for example, silylated versus non-silylated end glycans, or other types of structures, fucosylations versus non-fucosylated structures. We will do the same thing for O glycans. We can release these using chemical methods rather than enzymatic methods, and we get a profile of O-linked glycans. O-linked glycans are less complicated, but they're usually a little bit harder to make sure that we get all the glycans. Because they're smaller, we have to be careful we don't lose them through our isolation, purification, or derivatization procedures. So there are different, like tweaking the methods always to make sure that you get the best data out of the cells or tissues that you're looking at. We do the same thing looking at silylated or non-silylated glycans within the O-linked glycans, and again, fucosylation and other structures that may be present. Once we are armed with the glycomics data and we know what type of glycans are present, we also want to know what are the proteins that are present, what are the glycoproteins that are present, and what is the site of glycosylation. So we want to know what is the peptide that is attaching these glycans. Each glycopeptide site can have multiple glycans attached, and we want to have that information in order to compare glycoproteomes of different cells or tissues from each other. More and more people are now interested not just in the glycomics, but they want to know what are the glycoproteins that are present and what are the glycan sites. So we digest the protein, as I said, and we will try using the information we had our glycomics to try to determine the peptide with the corresponding glycans and their relative percentages. This is an example of that, a particular peptide having the relative abundance of the glycans attached to a single peptide. Using optimized fragmentations, we can really also start to deeply looking at the glycans that are attached at these sites. For example, this is an example, the top one is an example of the alpha acid glycoprotein from a trypsin digest that we're looking at. This is an HCD fragmentation compared to a more optimized, newer fragmentation methods. UVPD fragmentation are our newest instrument. That now we're looking at the positions of the fucose on the glycans. And we can tell the fucose being attached to the core or on the antenna. So that kind of information really allows you to go more deeply into the structural information once you have the glycopeptide analysis. Also, non-carbohydrate constituents are important attachments for the glycans. And here is an example of the UVPD giving us more information on the position of the acetate group. There's two acetate groups present on this glycan attached to the peptide in erythropoietin, and the UVPD really allows us better fragmentations of the glycans. So we are excited in applying the UVPD to a lot of glycoproteomics data moving forward. So with that information, I'm going to give you an example of how we went about looking at the spike protein and its human receptor. And also since then, since 2020, we've done quite a lot of variants, spike protein variants, and I want to share that data with you. So just a little bit introduction about the SARS-CoV-2. So at the beginning of the pandemic, we and others started really opening our labs for the SARS-CoV-2 research. And a lot of work has been done since then, and now it's been determined that the spike protein attaches to the ACE2 receptor on the surface of the human cells, and glycosaminoglycans act as a director and help mediate the attachments of the spike protein to the receptor binding region, the mediators of the receptor binding regions of the ACE2, and it helps the interactions. So basically, once the ACE2 and the spike protein are attached to each other on the cell surface, the virus moves in, and there is the series of reactions that happen. The viral genome is released, and the RNA is translated into proteins through the host ribosomes. And later on, the spike protein and the different type of proteins, the envelope protein, nucleocapsid proteins, and the membrane proteins are formed using the host machinery systems. They go through the ER and the Golgi, they get glycosylated and they get processed, and the viral proteins combined with the nucleocapsid protein vesicles are formed, and then the newly synthesized viruses leave the host cells through exocytosis. So back in 2020, when we first started looking at the spike, we were interested in the spike protein glycosylation. We initially looked at there is two subunits in spike protein, the S1 and the S2. We originally looked at these separately because we thought maybe when the vaccines companies want to use it, they may only use one subunit versus another. So we looked at these originally in different subunits separately from each other. So when we looked at the glycosylation, so we did Glycomics and Glycoproteomics, and when we looked at the glycosylation of the spike protein, we found varying degrees of N-glycosylations in both the S subunit and the S2 subunit. We showed that some sites were had high manostructures, that's the green structures, and the yellow structures are the ones that are complex types. Some sites, when we looked at these separately, were not glycosylated, especially in the S2 subunit. Interestingly, our group was one of the first groups that reported on the O-glycosylation being present near the RBD regions, in the flexible regions, as threonine 323. The amount of O-glycosylation since then has been studied extensively. More O-glycans were later found, up to like 17 different sites have been found. And it all depends basically on where the S protein is being isolated from, which kind of cell lines is being used, what conditions is being used, and whether it's from actual human cells or insect cells. So that degree of glycosylation as a whole, and specifically O-glycosylation, is very sensitive to those conditions that the cells is being grown. So we found both core 1 and core 2 type O-glycans on the S protein S1 subunit. Interestingly, the N165 and N234 was later found out to be important for the binding, and those are important for having the up conformation when it's being faced with the ACE2 receptor. So this is a summary of the N and O-link glycosylations of the SARS-CoV-2. As you can see, the S2 subunit is heavily glycosylated, a lot more complex types, whereas the S1, we did see a couple of the sites, especially 343 and N331 being heavily with high nanostructures. Once we had this study, we wanted to look into more the ACE2 receptor, the human angiotensin converting enzyme. So this is the structure that shows the SARS-CoV-2 spike RBD region interacting with the ACE2 receptor. So there is simultaneous binding of the two S protein trimers to an ACE2 dimer. Some of these N-glycosylation sites that I'm showing here have been shown to be important for that binding, especially the N322 and the N90. So we set out to look to see which species are susceptible for and which species are not susceptible for SARS-CoV-2 infection. And we compared the glycosylation sites with that susceptibility. We found that this protein structure and glycosylation sites alone are kind of inconclusive, except for N90. N90 has been shown to be important for if the species is susceptible. For example, pig is not glycosylated. The ACE2 receptor in pig is not glycosylated in N90 for pig, mouse, and chicken. And the others that are susceptible are glycosylated at N90. So we performed comprehensive glycomics analysis of the ACE2 receptor, and we found out that these were expressed in HEK293 cells, and they were heavily glycosylated with lots of complex type glycans, fucosylations, and also up to 3 sialic acids, significant amount of still no sialic acids, and some with one fucose. The predominant structures with biantennary, with triantennary and tetraantennary structures, including hybrid types. We looked at there were some bisecting glucanacs, and we also looked at the sialic acid linkage using MS analysis and found out that 2,3 linked sialylated N-glycans on the ACE2 were the predominant linkages. Maybe they are favored the binding of coronavirus S proteins. So we also performed glycoproteomics analysis, and interestingly, we used the full length ACE2, and at the time, a lot of people were also looking at the ACE2 glycosylations. Since we used the full length ACE2, we were able to report the first structures on the O-link glycans. The O730 was fully O-glycosylated here, and we detected 7 N-link glycosylation sites on the ACE2 glycoprotein, and some were heavily glycosylated. Most were, all were fully occupied, actually. So with that, basically the presence or absence of glycosylation in certain positions of ACE2 near the binding sites does not seem to correlate with susceptibility to SARS-CoV-2 infection, except the N90 glycosylation occurs only in susceptible species. However, in humans, absence of N90 glycosylation is associated with stronger binding between spike and ACE2, while the N322 enhances the binding. The glycan structure, so the glycan structure may be rather than the presence or absence may be critical for that kind of binding, the particular glycan structures that is present. Interestingly, children express more ACE2 than adults, but adults are more susceptible. And ACE2 was seen to drastically decrease with aging in rat models. So with that, we started looking at different variants of the SARS-CoV-2 spike protein. This is showing the different mutations that are present in spike protein. As you can see, initially in 2021 and 2022, we looked at the alpha, beta, gamma, delta, and omicron. As you can see, the omicron obviously has a lot more mutations than others. And we looked at both glycomics and glycoproteomics of these variants. As you can see, this is an example of the N-glycomics and the O-glycomics data of these different variants. Moving from the wild type through the alpha and the different forms of the mutations, the last one is the omicron, you can see that the level of high nanostructures is increasing as we go through these mutations. Obviously, the omicron has the most mutations and has the highest levels of high nanostructures. Also, as you can see, the fucose and the sialic acids is also decreasing, obviously, with the Omicron species, the Omicron mutations. The O-glycomics, the interesting about O-glycomics is they're all glycosylated. There's one missing O-glycosylations. They're all O-glycosylated, showing that O-glycosylation is important. There is different variations in the levels of O-glycosylations, but the level is so low that I think that the quantitations, I wouldn't bank on the quantitation of these because they're maybe about 1 to 5% O-glycosylations, but it's consistently present. We later looked at the sites of glycosylation of these variants, and what you can just see in this slide is, what I can tell you is carbohydrate glycosylation and spike protein is pretty consistent. It's interesting that moving from all these variants, even the Omicron, it's pretty consistent that you can see. I'm just showing the main glycans that are present. Of course, each site had lots of different glycans. I'm just showing the main glycans, but moving from one to next, it's pretty consistent. But both the glycopeptide and the glycomics analysis showed that the Omicron stands out as compared to the others. All the sites were listed as one variable, and then the PCA analysis was performed. Omicron, as I said, showed the highest variance compared to the rest of the COVID-19 variants. Later on, we moved to other variants across Omicron. The ones that I have circled are the ones that we've looked at more recently. We've looked at 7 additional variants since we looked at the last Omicron in 2022. So in September 2022, this XBB 1.5 variant came along, and it showed enhanced transmissibility and immune invasion. And later on, that XBB 1.5 was replaced by the BA286 and the GN1. It was basically replaced all others. And interestingly, this BA286 and GN1 variants have created 2 new N-link glycosylation sites as positions N245 and N345. We know that viruses over time, is maybe 50 years or 100 years, they increase their glycan shield. And in the case of SARS-CoV-2, within like 5 years, SARS-CoV-2 is adding more glycosylation sites. So these more recent ones have 2 additional glycosylation sites. And studies have been shown that the N354 glycan stabilizes the spike protein trimer in the RBD down conformation, potentially impacting viral infectivity and pathogenicity. So what does the Glycomics analysis of these Omicron variants show? They show varying level XBB lineages. Basically, they're very similar, except maybe moving the high nanostructure is increasing slightly as we move along. However, the BA lineages, if you take a look, the GN1 stands out because it has the least amount of high nanostructures. So this GN1 that has the extra 2 glycosylation sites also is showing basically more complex type glycans and more fucosylations, more sialic acids. This is the distribution N glycosylation sites within the spike protein domain. And again, the GN1 stands out both in its N-terminal domain, the RBD region, and the C-terminal domain as being has the least amount of all oligomanostructures. This is the glycosylation of these N245 in the 2 different BA2886 and GN1. As you can see, again, the GN1 is showing less high nanostructures at N245, whereas the BA286 has 17%. And this is the N354 that the GN1 really has about only 1% of high nanostructures. This is the PCA analysis of Glycomics of the variants, showing the GN1 and the BA286 really standing out and being very different to the others. So the glycosylation is really changing over time, whereas the sites of glycosylation is not. All of it still remains glycosylated, but the actual glycans are changing in response to different things. So in summary of the SARS-CoV-2 data, almost all sites except the N17 in Delta that is missing, the majority of abundant glycans are almost conserved across all the variants. In most cases, the most abundant glycans are the same in variants. Our study on the comparison of the glycosylation patterns of SARS-CoV-2 variants shed light to understand how virus evolve itself to trick the host immune system. But an interesting thing is that the question still remains that how SARS-CoV-2 virus preserved both N and O glycosylation sites across all these different spontaneous mutations, and so indicating that glycosylation must be critical for its survival and for its activity. I want to switch gears and talk a little bit about glycolipids. My lab in the last 5-6 years has done quite a lot of method development for looking at high sensitivity MS methods for identification and quantifications of glycolipids. So we've used 2 complementary methods to characterize glycosphingolipids. Historically, when we start from cells or tissues here, we do a homogenizations and we do an extractions to get the glycolipids away from the protein fractions and other lipids. We do a saponifications and basically we separate out the glycolipids from the free lipids and the proteins. Once we have the isolated glycosphingolipids, we have developed 2 kind of complementary method. One is a method that is more historical and it's been used traditionally, is where the intact glycosphingolipid is permethylated, and we looked at it by mass spec, MALDI, or electrospray. Here, we've decided to cleave the glycans away from the lipid. We've used the endoglycoceramidase digestions to cleave the glycans, and we've tagged the glycans with a prokinamide and then looked at those by LCMS. We can then later look at the lipids separately. We can look at those by mass spec using electrospray or GCMS and be able to look at these different populations. So this is an example of the first approach where we permethylated the intact glycosphingolipids, and the benefit of the permethylation is boost sensitivity and ionization in electrosprays. It allows for better detection and sensitivity in the MS2 profiles. So this is an example of the lipids being attached to the glycans. We use the MS to look at the finer structures, get more structural information on the glycan portion of the glycolipids. The second method, as I said, is use of ceramidases, endoglycoceramidases to cleave the glycans. We separate those by C18, and we did a prokinamide basically derivatization. The prokinamide gives you sensitivity and it allows for separation of these glycans using LCMS. This is an example of results from our prokinamide, results of the ceramidase digestions. So signal accumulates for GSL with similar head groups but heterogeneous in the ceramide. So it helps reduce the complexity of the data because now the glycans are separated from the lipid. It allows for separation of the isomers thanks to chromatography separations. So we've done, we've looked at different degree of silylation and fucosylations, and we've been able to quantitate the glycans and look at, we've looked at the lipids by mass spec and by GCMS. This is the comparative data between the permethylation and the enzymatic cleavage. So with the permethylated glycosphingolipid, we were able to see 24 glycosphingolipids from 7 different glycoforms. We were able to get information about the ceramide and the glycan. The data was complex for data interpretation, and to quantifications, we used internal standards. With the enzymatic cleavage and labeling, we were able to identify 18 masses of glycoforms. It was glycan focused. We signal accumulations for GSLs with similar head groups. We increased sensitivity, and the fluorescence allowed for quantifications. So we were able to see a lot more of the glycans, especially the larger glycans, that we were not able to see with the permethylations. So this is a complementary method that can be used. Prokinamide is a pretty versatile derivatization method that can be used to accurately quantitate the glycans that are present in glycosphingolipids. So both methods are useful for different applications. With that, I just want to go through a couple of collaborations that we've done and give you an example of the flavors of the kind of work we do with different people. This is an example of a collaboration with Simon Winovsky from University of British Columbia. He was interested in glycosyl transferase ST3-GAL4, which drives the immune invasion in acute amyloid leukemia by synthesizing ligands for glycoimmune checkpoints receptor CCLAC9. So immunotherapy based on protein-protein interactions has demonstrated promise for AML. In our study, the focus was on the role of silylated glycans in driving immune invasions of AML cancer cells. So AML cancer cells express ligands for CCLAC9 binding glycans at a particular high levels. And the ST3-GAL1 glycosyl transferase appeared to be responsible for this elevated CCLAC9 ligand expression in these cells. And the CRISPR-mediated S3-Gal4 knockout alleviated expression of the CYcLAC9. So we performed comprehensive glycomics analysis on aniline glycans, as you can see here, and this work implicated that S3-Gal4 as a potential target for immunotherapy in AML1 cells. This is an interesting collaboration with Jennifer Kohler. We've had a long-term collaboration with Jennifer from UT Southwestern. And this showed actually very interesting study showed the fucosylation of glycoforms and glycolipids. They have opposing roles in cholera toxin intoxication. So cholera toxin is an agent of cholera infections. Multiple classes of fucosylated glycoconjugate function in CT bind and intoxication of the epithelial cells. So interestingly, the knockout B3GNT5 has been shown to reduce the CT binding, but synthesizes the cells to intoxication, acts as a decoy. And the knockout of B3-Gal-T5 causes increased production of fucosylated Orlin glycoproteins and leads to increased CT binding and intoxication. So distinct classes of fucosylated glycans have different functions and regulate CT in different ways. So our data was consistent with the model in which relative expression of fucosylated glycosphingolipids and fucosylated glycoproteins control the extent of CT sensitivity. So we use this study as a model system to test our procainamide analysis. And this proved helpful because we were able to see some higher molecular weights fucosylated structures using our ceramide derivatization methodology. This has recently been published. I really didn't have time to talk about all the work that we do with the polysaccharides. My lab, probably like 40–50% of what we do is polysaccharide work, specifically a lot of work on bacterial polysaccharides. This is a recent study, interesting study of how bacteria compete with each other and what do they do in this competition. This study was a collaboration with Talia Karasov from University of Utah that was published this past summer. These phage-like bacteriums, they called tail coins. These tail coins are present in bacteria, and they basically kill other bacteria, other competing bacteria, and they form this competition. And they wanted to study these tail coins better. This is an example of Pseudomonas, and this was actually present in plants, but these are being used, the study is being used in looking at these tail coins for other bacterials also. So these tail coins are present in Pseudomonas, and they kind of kill, they're the cause of killing other bacteria and causing this competition. So we looked at, they wanted to find out what is responsive for these tail coins. And they looked at the gene studies, and they looked at the genes that were responsible in susceptible versus the ones that were resistant, and they found that the genes were present in the antigen regions of the LPS. We isolated the LPS of susceptible versus resistant strains, 2 resistant strains versus 2 susceptible strains, and we found out that the resistant strain lacked this O antigen, high molecular weight O antigen. We showed that using both SDS and then characterization. We found out the high molecular weight ramenose O antigen was basically the resistant one lacked this O antigen. The study is ongoing. We're still collaborating with her, and we're doing more detailed structural elucidations of the sequence of that high molecular weight or antigen. With that, I want to talk to you a little bit about our training program. As I said, this year is our 40th year. We are offering hands-on training courses. I think the CCRC has been one of the first groups that were offering these kinds of facilities and detailed structural characterization and isolation of polysaccharides and glycoproteins to the scientific community for a long time. So our hands-on training courses involve 4 courses: techniques for characterization of carbohydrates, polysaccharide course, microbial and plant polysaccharides. These usually run in the summer. Separation and characterization of glycoproteins and glycolipids. These are 1-week courses with 4 days of lab work and 1 day of module. You can choose from modules, mass spec module, molecular modeling modules, or NMR modules. Back in 2007, 2008, we started offering analytical techniques for structural characterization of sulfated polysaccharides, particularly glycosaminoglycans. So participants learn how to basically do molecular weight determination of glycosaminoglycans, disaccharide analysis, NMR, and mass spec of glycosaminoglycans and other sulfated polysaccharides. For the last 10 years, we've offered this bioinformatics course, actually in collaboration with scientists from Glygen. It's targeted towards mass spectrometry of glycoproteins. And so they go through glycoproteomics and glycomics data and the bioinformatics tools that are present in doing this data analysis. Most of our faculty at the CCRC are involved with lecturing and being involved in our courses, all 4 of our courses. Since the pandemic, we're now also offering 2 virtual free courses. One is Fundamentals of Polysaccharide Analysis, and another is Fundamentals of Glycomics and Glycoproteomics, and actually some GAG analysis. So those are usually in December. You can sign up for these courses on our website, and you can email me if you have any information. If you need any information about our courses, and we also do, we are going to start doing more we call it scientists in residence as part of the BioGrade NSF grant that we are involved with. We're going to have scientists come to the CCRC as part of BioGrade and stay maybe 2 weeks, 3 weeks, 2 months, 3 months with their own projects and come and use our facilities, our mass spec facilities, maybe developing high throughput glycoanalytics, maybe using our enzymes. We have probably one of the largest array of glycoenzymes here for structural elucidation. So stay tuned for that information. If you have projects that you think needs our facilities here and our expertise, we can arrange for you to come here, stay extended amount of time, and really take a deep dive into glycoscience at the CCRC. This is a geographical distribution of our service, training, and really our collaborators within the United States and around the world. The blue dots are for training, and the red is for really service and a lot of collaborations that we do. We are involved with, as I said, both service and collaboration. So depending on the scope of the work, some projects can take a couple of weeks, some projects take a couple of years. So it all depends on the extent of your work, what it involves. We work with both government agencies like FDA, US Pharmacopoeia, USDA, and a lot of universities, biopharmaceutical industry, and biotech industry. With that, I want to really thank our group that we have a great team at the Analytical Service and Training Laboratory, and my colleagues at the CCRC helping us really all through all our projects, really help us to get the best analytical method out to our collaborators. I want to particularly thank Christian Heinz, who's been with the group and really co-directs the group with me, and a lot of our senior scientists, Stephanie Archer-Hartman, Arthur Muczynski, Ian Black, Lee Tan, and a lot of others who really manage the group, and they're on the ground doing a lot of work. I want to thank our funding. As I said, I'm the PI on a national glycoscience resource, our NIH. Most of the work that I have done, to talk to you today was through our national glycoscience resource. I'm part of the Glycomip, glycomaterials, NSF grant, PI on the DOE Center grant, and more recently involved with the BioGrade, and a lot of good things are going to come out of the BioGrade, and stay tuned for a lot of our training components, and whether it's hands-on training here at the CCRC or residents in coming here for a long period of time, and a lot of our high throughput and enzymes being available for the scientific community. And thank you for your attention.